Yeah, thank you very much, Malte, and thank you for all of you to be so many here to listen to this educational session. I will have a very precise approach today, talk about some detail onto how we can use CRISPR to mimic human mutation in mouse, and I will have a special focus onto how we can engineer structural variants in animal models, and more particularly in mouse. So as most of you already know, structural variants are impacting the structure of the genome, the linear structure of the genome. So here we can see a particular loci with four different subloci, A, B, C, and D. And structural variant comes in different flavors. They can be an insertion of this small new loci, number E here. It can be a deletion in the second part of the loci B, an inversion where B and C are inverted here, a duplication of a fragment B, or a translocation between different chromosomes. All of these structural variants can lead to various effects, and the big advantage of having different technology to be able to mimic them in the mouse is to understand through which way they can impact the normal function of the genome. Structural variant can be tested routinely by diagnostics using RACGH, and they are responsible for a very large amount of inherited or herited disease, such as intellectual disability, but also developmental malformations. There is, of course, a very strong interest in being able to produce animal model to be able to predict the pathogenicity of structural variants. Every one of us here have some kind of a structural variant in our genome, although most of us are very healthy. So it is important when we have a patient genome to be able to direct our assay to one of the particular variants that we think is pathogenic. And so far, the method that were available to do that were pretty reduced. The most common used one is radiation-induced, where, for example, this mesomelic dysplasia could be mimicked by producing a large duplication, if I remember correctly, at the Hox locus. But the problem with irradiation is that it is a very random process. There is a lot of mutation induced, but it's completely random. A way to be more directed and more precise about it was to use the CRILOX technology that comes again in different flavors here. This means targeting two LOX-P sites, which are pretty reduced sequence, around 30-40 base pair, at two different positions in the chromosome. By adding on top of it a Cre recombinase, it is possible to induce a recombination that would delete the intermediate region. If the two LOX-P sites are in a reverse orientation, they will induce an inversion of the intermediate sequence. An extended step of that it was the possibility to have the LOX-P site on different chromosomes. And by inducing a strong recombination, it is possible to induce a translocation between the chromosome, leading to an unbalanced translocation with, on the one side, a deletion, and on the other, a duplication. With this, it was possible to produce, to mimic a human condition, the Smith-Magenis syndrome, for example, and many others. However, this system is here extremely time-consuming. You need to target the first LOX-P site, the second LOX-P site, cross the mouse with a Cre driver mouse, produce your line, find your animals, and so on and so forth. It's taking forever. But now, the revolution has come with the CRISPR Cast technology. And as Greg has told it before, the Cast system, CRISPR Cast system, is pretty handy. It actually needs two components. One is the Cas9, which is an endonuclease which cleaves the DNA in double-strand break. And the other is an RNA guidance system, which on the one side bind to the Cas9 endonuclease and bring it on this other side to a specific genomic region. Originally, these systems come from bacteria, and these bacteria use it to cleave and destroy the DNA of infected viruses. It was, in the recent years, adapted in mammal cells, mammal embryos, to produce mutations. So I would say the CRISPR-Cas is simple, precise, fast, and really inexpensive. Again, as Greg said, the Cas9 system itself does not produce the mutation. What produces the mutation is the way the cell is trying to repair the DNA. And it can do it through two different ways. The first one here is the homology directed repair. In this case, the cell will look for a template DNA, which it will try to copy in order to fill in the gap where the double-strand break occurs. And this can be taken advantage of to introduce specific cassette like GFP or LAG-Z markers to understand the expression of a gene, or to introduce specific variants to change the coding sequence or whatever specific nucleotide you want to integrate. On the other side, the non-homologous end joining is another mechanism. And here the cell will not look for a template donor, but will just ligate the two DNA ends, which are now not together anymore. And by doing this, it will introduce pretty frequently mistakes, errors. And this will produce then a possibility to introduce mutations. Now, both of the pathways are targetable by drugs. So we can force the cell to prefer non-homologous end joining or homologous directed repair by just adding different drugs here. If you want to produce a structural variant, it's a slightly different story. You need to, instead of using one single targeted guide RNA and a Cas9, you just use two of them. And they can be very far from one another. They can be hundreds of kb away. Actually, we already produced deletion of around 2 megabase of DNA. And so by bringing these two guide RNA at different positions, they will cut at two sites, produce a double-strand break, and the cell will start to repair it in different ways. First possibility is that nothing happens. So the allele stays pretty much wild type, and you will have mutation occurring at one of the targeted sites and at the other targeted site. The other option is that the cell will just kick out the intermediate DNA and produce a very large deletion, like in this case here. It can also produce an inversion of the intermediate sequence and a duplication. This duplication most likely is coming from the fact that it produces an unbalanced translocation between two sister chromosomes. Right. So I will go through the way that we can produce this structural variant. It's a very simple and fast procedure that takes, let's say, 10 weeks when we do a little bit of advertisement, maybe 2 months, or maybe 3 months or 4 months when you're actually really doing it in the lab. So it starts basically with designing your guide RNA and ordering it. It's very quick, and then you introduce it into a vector that contains the whole guide RNA guidance systems plus the Cas9. Then this guide RNA is transfected into mouse ES cells, pluripotent cells, and cells are submitted to a strong selection just to remove all the cells that didn't get any of the plasmid. And these cells will then grow and produce some small colony of around 20, 30, 40 cells that you can pick and select and isolate into a 96-well plate. You grow them, and then within this 96-well plate, what you're doing is just looking, using a very simple PCR approach for the structural variant that you're looking at. For example, a deletion here. So in your 96-well plate, you will have a certain frequency of these alleles that occur. Then you select the specific one, you expand your cells so that you have a very big amount of cells, and you can produce a mouse out of it. Now we'll go into detail into the different steps that allow us to do that. So the first one is obviously to design a good guide RNA. So how do we design a good guide RNA for this particular purpose here? Here I show something, a tool, an internet tool that is actually pretty handy. That is called BenchLink. It's very nice, and you have actually all the data directly on it, and this is how it presents. Here you have a little piece of chromosome 1, where we are interested to design a guide RNA. And you can see here on the left that a lot of different information appears, right? First, here is just the position of the guide RNA. This is the guide RNA itself. And as Greg mentioned, this is the PAM sequence. The PAM sequence is not encoded into the guide RNA, but it's the place where the Cas9 will cut. So if the guide RNA hybridizes somewhere and doesn't have a PAM sequence, it will not cut. Then there is an on-target score, right, which is interesting to know because it tells you how the Cas9 system will be targeted, with which strength it will be targeted to the region. And there is an off-target effect, score, which is also extremely important because it tells you how frequent there is a chance that the Cas9 endonuclease will cut or hybridize somewhere else in the genome. And that's something that you really don't want. If we look in detail here, we have our first targeted region here that has absolutely no mismatch in DNA. You have the PAM sequence and the score. And then you have all these guys here that are possible off-target effect. You see that they have mismatches, like 2 for the first one, and they also have a PAM sequence, and they have a score. So here, when we design this guide RNA, we are just careful about a few factors. First, of course, that there is a PAM sequence. You can see here at the bottom of the list, we have some pretty nice off-target, but they don't have a PAM sequence. They have an AG, and you should have an NGG here. So these one are no problem. So the rule that we have is that we try to avoid mismatch. It's not so important if there are mismatches at the 5' end of the guiding sequence because they are more tolerated than others. If off-target effect, predicted off-target region has more than 3 mismatches, it's not so important. It will completely inhibit the off-target effect. A score of lower than 1.6 is our threshold. So, for example, this guide RNA is not good because the first one has the first off-target here has a score of 1.8. And of course, what you want to avoid is coding sequence. So if you have an off-target in the middle of a repeat or something, it's not so important. If it's an off-target effect that occurs in a gene, then it might be a problem. How do you genotype your clone? Is very, very handy and super easy. Just design four primers, one on every side of the first cutting site and one on every side of the second cutting site. When you have a deletion, you just do the PCR with this primer here and the one on the other side. When you have an inversion, you do this combination. So it's a very trivial process here. And just by doing this, migrating the PCR on the gel, you will have all your flavors. So, as I said, we cannot control from the design if we will obtain a deletion, if we will obtain an inversion or a duplication. So we need to genotype every clone for all of these possibilities. What you will most frequently have is a wild-type clone where we don't see any of the structural variant in between. But sometimes you will have just a deletion or just an inversion. Sometimes we'll have pure duplication. But often what happens is that we'll start to have several mutations. So on one allele, a deletion, and on the other, a duplication. This maybe you're not interested, or maybe you are. And this is the advantage of the system here. It produces a lot of the structural variant in one shot. The other stuff that's very important to check is what happens at the targeted sites. Ideally here, in a deletion, this is the sequence that we should have if the cutting was extremely precise. However, the cutting is not precise and induces a lot of indels. So we obtain here the small deletions that are different in every deletion clone that we obtain. Same is true for inversion, same is true with duplication. But also, what happens is that at the wild-type breakpoint, if you have a clone that you think is wild-type, like this one here, you will see that actually at the targeted site there are mutations. Because CRISPR is so efficient, it will systematically produce a mutation, maybe not the structural variant, but just a small indels in the wild-type sequence. When multiplexing, if you want to produce several regions in parallel, so you target different chromosomes with your Cas9 system, you have to be careful. One thing is chromosomal rearrangement. Chromosomal rearrangement have been shown to be inducible by targeting 2 different guide RNA, 2 different regions. So, of course, again, here you need to test for that, and you need to sequence the cutting site in order to be absolutely sure that you have a minimal amount of target and of undesired product. This is a bit old slide. It comes from a few years ago when we started here. We have seen that at every locus that we tried here, FRNA4, LAF4, PEDX1, Indiana jog, BMP2, or H2FI, we have been able to produce deletions or inversions. In some of them, it was a bit more difficult to obtain duplication. But nowadays, we have been able to produce it almost systematically by just changing a guide RNA somewhere if it doesn't work, whatever mutation we wanted. Another thing that can help a little bit your design is to design your guide RNA into open chromatin regions. These regions, which are depleted in nucleosome, are much more easily targetable by a Cas9 system. How do you produce a mouse then? Then there is several possibilities here. if you have your ES cells, the most easy one is to take a blastocyst or a 3.5 days mouse embryos and to inject directly within it your modified embryonic stem cells. You transfer this blastocyst back to a foster mother, and then you obtain a chimeric mouse that is partially wild type and partially engineered mutant. This is good, but again, then it means that you have several steps of crossing that you need to go through, and that's maybe not what you want. You want to be fast, and you want to have a limited animal cost, of course. And so the other option is that when you have a 2-cell stage zygote, you apply a slight electric shock to it, and this will induce a cell fusion and result in a tetraploid cells. This zygote will develop until a certain stage and eventually dies because a tetraploid mouse doesn't exist and doesn't survive. However, if you just bring in this embryonic stem cells that have been modified, that are not tetraploid, it will rescue the blastocyst, and it will make a mouse that is entirely derived from these modified cells. And in that case, no need for a line. You can directly look at your embryos, look at a young mouse, and in a homologous fashion. So in that case, it's actually extremely efficient. I would like here to pinpoint out that recently people have published the possibility of producing structural variant directly by injecting the CRISPR-Cas9 system and the guide RNA into the mouse oocytes, into the mouse zygotes, and this will produce directly offspring which are mutated. Unfortunately, of course, every offspring will have a slightly different mutation, and maybe in that case, you again need to look at your mouse and produce mouse lines. Now I'll go into a few examples on how we can apply this approach and how it was so crazy and so nice for us to have it. So this is a story that started maybe 2006 or something like that, where a patient had a triangular tibia and a fibular aplasia, which is a very classical, which is a syndrome that was at the time novel, and we didn't know really much about it. So we went to a RACGH, and we could find that this patient had a deletion in a gene called LAV4. And when we did an in situ hybridization to see where LAV4 was expressed, we saw that it was actually expressed in the perichondrium. So you see here in this mouse hind limb. So we were really happy at the time and saw that everything is super clear. This LAV4 is the cause, the loss of function of LAV4 is the cause of this triangular tibia, and so on. So we produced a knockout allele, just kicking out the gene, and unfortunately, nothing happens. The tibia is here absolutely fine. There was absolutely no problem, and that was taking a long time because we needed to produce a knockout of the mouse and so on, and analyze the mouse and so on. So the student that was doing it at the time decided to go back to the original data and map the mutation in a better way. And what she found was pretty interesting. She found that actually deletion was not a complete loss of function of the gene, but rather a partial inframe deletion. The deletion started in one of the intron, in intron 2 of the gene, and stopped in intron 9, removing 7 exons of the gene and leaving a partially inframe protein. And this is different, of course, than a total loss of function, because we have a remaining protein that could act, for example, as a dominant negative and remove the effect of other genes. So, with the help of CRISPR-Cas9, she produced this allele directly homozygotally in the mouse, and that was very efficient, and she directly could see that indeed, when you do that, you obtain exactly the same mutation that you have in the patient. So you have here a triangular tibia and a fibular aplasia. We don't see anymore the fibula. And also some minor characteristic of the patient were reproduced, like the defect in the pelvis. So this is one way that you can do it, and the other way that we are in the laboratory more interested in is how this affects gene regulation, how this structural variant can change the expression of a gene during development. To understand that, you have to, of course, see the genome in a 2D way. Most of us see the genome as being containing genes on one side, regulatory element on the other, but of course this genome is packed into the nucleus, and it's forming into a 3D structure through a process that we call chromatin folding. The chromatin folding is, of course, at the level of the whole nuclear, but also at the level of single gene. Single gene are able to contact distal regulatory elements that are sometimes located hundreds of kb's away. And the precise timing and architecture of this allows for the normal transcription of genes and the normal patterning of our body structures and also a normal homeostasis. One of the main features of the 3D structure that was shown to exist a few years ago is called topological domains. So these domains, here represented in red, are regions of preferential interactions. So the redder here means that the two regions interact more. For example, this point here shows that this region and this one are actually interacting frequently together. When it's white, it's not interacting. So what I want to say here is that you can see that this small locus here is compartmented into 3 regions of preferential interactions. These regions are called topologically associated domain, TAD. They are pretty much conserved across different cell types and also to some extent between species. And one of the most important effects of TAD for gene regulation is that they actually facilitate the enhancer-promoter contact, right? So this enhancer that you see here will not screen the whole genome to find its target gene, but just its own topological domain and find the gene A pretty quickly and activated it. TAD are also separated by boundary region, right? And this, of course, leaves us with the possibility that structural variant may influence this organization. And I will show you how they can do it. And to do that, I will go to a specific locus here called the E02 locus. So in the E02 locus, you have 2 boundaries. Here, on this very large TAD, you have the E02 gene and you have enhancers. And what we know is that a couple of structural variants at this locus have been shown to result in limb defect. So the patterning of the limb was wrong. This deletion here produced brachydactyly, so it's a shortening of the fingers. The inversion here produces the F syndrome. And finally, duplication here produces a polydactyly, so extra digits. And how does this work? Well, to understand how it works, we used the system that I just described. We engineered the same alleles as the human patient had into the mouse. And what we could see is that the mouse directly displayed very similar phenotype. Here, brachydactyly, shortening of the fingers. And this is a very good model because now we have the mouse that mimics the system, we have living animals, and we can now understand what pathomechanism brings us from the structural variant to the phenotype. And this we can do, for example, just by starting with RNA-seq. So on the top track here, you have the RNA-seq of the region. You see that some genes are expressed, some others are not. But in the mutant, this gene, PAX3, that is here in the middle, is the only gene, the locus, that gets overexpressed in a very specific manner. And if you take just this gene, PAX3, and you overexpress it in transgenic embryos, you obtain again the same effect. So first, we could see here which gene is responsible for the congenital defect. But moreover, it also was important to understand how could this gene be more expressed in the case of this deletion? What happened exactly? So you see here, this is the normal expression of the E02 gene, which is here. It is actually pretty distal expression. It is actually controlled by a cluster of enhancer that display a very similar expression activity, also in the distal limb. Right. And the PAX3 gene that is just here is actually not expressed in the same pattern. It's a little bit expressed in the proximal part of the limb. So the E02, the enhancer that are here are interacting, as you can see here all along this line, very strongly with the E02 gene that is here. But in the deletion, something different happens. Now, of course, the E02 is deleted, so there is no expression of the E02. And we can see here that the PAX3 genes start to have a very different pattern that normally has, right? That's the normal condition, and here it's an overexpression. And we see that this expression somehow looks a little bit like the one of E02, very distal, and so on. And if we look at our enhancer cluster here, we see that now it forms new interactions. Specifically this one, which is exactly with the PAX3 genes. So this deletion actually rewired the enhancer was the PAX3 gene. Basically, if we would look at the real map by removing the deleted part, this is how it will look, right? Normally, we have two topological domains which are insulated from one another, and here we just have one. So a little bit like if we would have put the enhancer and the gene in the same box, and now they can talk to each other and activate each other. Right. As a control, we also produce another type of allele, and this is also something that was very interesting in the field, is we produce the same deletion, but we left the boundary element intact. And you can see when we did that, we didn't see any overexpression of the PAX3 gene. Actually, if we look at the contact, we saw that the contact from the enhancer was limited to the boundary region, and that we didn't see extra contact a little bit further away. Right. So here, in that case, by preserving the boundary element, we could still split the two TAD into different regions of interaction and shelter the regulatory information and the gene. So with that, I hope that I showed you here how important it is to understand the effect of structural variants. We know that they can be important to remove some exon of a genes, but they can also be important in changing the regulatory activity. I think there are many talks from the lab, including from Malte here, that will speak about how structural variant can impact gene regulation. They are all based on the technology that I showed you today. It's a pretty easy hand-on technology. You just need to have some good embryonic stem cells, actually. Right. So with that, I would like to finish and just advertise for some colleagues. The first one is a talk by Anya Vil in the plenary session today, which will talk about the enhancer composition and dosage in the control of a very important gene, Indian hedgehog, in the formation of bones. Also deals with structural variant here. Then there will be the talk of Malte on the competitive, or what's the name? Competitive session, I think it's called, about the regulation of PDX1. And finally, another talk by me, just before Malte, actually, in the same session. And with that, I would like to thank all the lab, especially Katerina Kraft, who started this whole CRISPR system in the laboratory and made it work very efficiently. Of course, I would like to thank Dario Malte, and here also people that helped to produce this very nice high-C maps, capture high-C maps, and all the lab and the funding, of course. And you for your attention. Thank you.